In this video, we'll be discussing the creation of the different vaccines and review the performance data of several that have been released or are about to be released. The primary target for nearly all vaccines is the spike protein because entry of the virus into human cells is facilitated by binding the spike protein to the ACE2 receptors. Antibody directed against the binding region of the S protein blocks binding to human cells, and then the virus is unable to enter these cells and grow. All vaccines express the full-length spike protein. An alternative target being used by a minority of vaccine developers is the membrane protein. Before we discuss the different ways vaccines can generate immune responses to the spike protein, we need to address the concern that this coronavirus will mutate and become resistant to the vaccine. SARS CoV-2, like all RNA viruses, has poor proofreading and commonly makes mistakes or generates mutations. The most recent example was the discovery in the United Kingdom of a mutant called VUI 20212-01. VUI stands for Variant Under Investigation, and 2020 is the year of its discovery, and 12 the month of that discovery, and the final number is the order of discovery of the different mutants. So this is the first mutant in the month of December. There are a total of 17 mutations in this virus, but the one that is felt to explain its increased ability to spread is the N to Y 501 mutation. That is a shift at 501 from asparagine to tyrosine. This mutation results in higher affinity binding to the human ACE2 receptor. Tighter binding should enhance the virus's ability to enter and infect cells, and epidemiologic studies suggest that it spreads 50 to 80% more efficiently. Because the vaccines use the full length of the spike protein, they are expected to stimulate antibody and cell-mediated protection responses at multiple sites along the S2 binding domain of the S protein. Therefore, a single mutation is unlikely to reduce protection. Now let's discuss the four major classes of vaccines presently under production. They are nucleic acid vaccines, 2 viral vector vaccines, 3 live attenuated viral vaccines, and 4 synthetic protein vaccines. Let's begin with nucleic acid vaccines, because the first 2 vaccines released fall into this category. Both the Pfizer, BioNTech, and Moderna vaccines are RNA viruses. They consist of full-length RNA message of the spike protein that is packaged in a protective lipid nanoparticle. Nanoparticles are injected intramuscularly, and then the lipid nanoparticles are taken up by immune processing cells, dendritic cells, and monocytes. Once the RNA enters the cytoplasm of the message, it is used to synthesize the spike protein, and then the protein migrates to the membrane surface of the cells, where B cells are signaled to produce antibodies against the protein, and T lymphocytes are induced to generate cell-mediated killer T cells that destroy the virus. The Pfizer-BioNTech virus is called BNT162b2, a randomized clinical trial, RCT, consisting of 43,000 subjects compared 30 micrograms of vaccine to control subjects who were injected with saline. Of the half who received the vaccine, 8 developed COVID-19, while 163 in the placebo or control group became infected. This represents a 94% efficacy. Here are the curves comparing the placebo upper line to the vaccinated. The vertical axis is the number of cases as a percentage infected, and the horizontal line, time in days. Within 10 days of receiving the first dose of the vaccine, you can see that the number of cases flattened in the vaccine group, while cases continued to accumulate in the placebo group. When it came to severe cases, there was 1 in the vaccine group, versus 9 in the placebo group. With regards to side effects, there are only 4 severe side effects observed, 2 with swollen lymph nodes under the arms, axillary adenopathy, 1 with ventricular premature contractions, a heart arrhythmia, and 1 episode of paresthesias, or temporary loss of sensation in the legs. As this figure shows, mild to moderate systemic side effects were more common after the second injection, fatigue and headaches being the most common side effects. 2 indicators that predict protection are the level of antibody generated against the spike protein and the level of neutralizing antibodies. As shown on the left, the Moderna RNA vaccine reached levels of 300,000 to 3 million ELISA antibody units. Neutralizing antibodies are shown in the right figure. Mixing of serum with virus kills the virus at dilutions of 1 to 300 to 1 to 400 in vaccinated subjects, but not in placebo. As noted with the Pfizer vaccine, efficacy was 90% for those receiving vaccine, and 100% were protected from contracting serious disease. Next are the viral vaccines. Both the Oxford AstraZeneca and the Johnson & Johnson vaccines have used adeno-associated viruses, abbreviated AAV. This single-stranded DNA virus has been used in gene therapy for over 20 years. The DNA message for the spike protein has been inserted into the virus, and when the virus is injected intramuscularly, it invades immune processing cells, where it generates messenger RNA for the spike protein that in turn guides the synthesis of the spike protein. Once synthesized, the protein migrates to the cell surface where B and T cells generate antibodies and program cells to kill the SARS CoV-2 virus. These vaccines are also in phase 3 studies, and preliminary analysis of the Oxford AstraZeneca vaccine have been published in Lancet. The trial consists of 23,848 participants from the United Kingdom, Brazil, and South Africa, and this vaccine, like the mRNA vaccines, require 2 doses. Control subjects received the meningococcal vaccine or saline. As you can see, the vaccine was protective. Compare the blue meningococcal vaccine recipients to those receiving the AAV virus in orange. This preliminary study demonstrates an efficacy of 71%. The third strategy being used is to create a live attenuated or weakened virus that has parts of its RNA deleted so it no longer can cause significant disease. The virus will be injected and is expected to induce immune responses without causing significant harm to those vaccinated. There is limited data on the vaccine, which is being manufactured and studied in India. Finally, the tried and true approach to vaccines is to inject the surface protein to induce an immune response. This approach has proved to be very effective for encapsulated bacterium, Strep pneumoniae, sometimes called the pneumonia vaccine, commercial name Prevnar 13, and also effective for Haemophilus influenza type B, abbreviated Hib vaccine. The Hib vaccine has nearly eliminated this once deadly infection in children. For the SARS CoV-2 vaccine, the full-length spike protein is expressed and purified and combined with an adjuvant, matrix M1 adjuvant, a component that amplifies the immune reaction and is injected intramuscularly, and the immune processing cells take up the foreign protein and express it on their surface, stimulating B and T cell immune responses. Early studies published in December of 2020 reveal that this vaccine induces a robust antibody response to both 5 micrograms and 25 micrograms of protein combined with adjuvant, and the levels are comparable to infected patients who required hospitalization. See the values within the black box, achieving levels of antibody of 47,000 to 63,000 ELISA units per ml. This approach also induces impressive viral neutralizing titers of 1 to 4,000 enclosed in the black box. A major question that remains to be answered is how long will immunity last following vaccination? This is a critical question to answer because if the administration of the vaccine is too slow, immunity for those who were vaccinated first may wane and they could again become susceptible to the virus, preventing the achievement of herd immunity. At the present rate of vaccination, it could take a year to achieve 60 to 80% coverage, and a significant percentage of those vaccinated early may no longer be protected. To answer this question, investigators looked at 21 individuals who were infected 17 years earlier with SARS-1, the close cousin of SARS CoV-2. They found that their T cells reacted to the nucleocapsid protein from SARS CoV-2, demonstrating long-term immunity. Examination of antibody levels following natural infection has been fraught with methodologic issues with regards to answering this question. We do know that mild infection induces lower immunoglobulin levels than more severe infections. As discussed in the diagnostic section, IgG and IgM, as well as neutralizing antibodies, reach detectable levels by approximately 14 days after the onset of symptoms. IgM levels decline over 3 to 5 weeks, while IgG levels and neutralizing antibodies levels peak in weeks 3 to 7 after the onset of symptoms, then plateau, persisting for at least 8 weeks. Systematic study of IgG and neutralizing antibodies remain to be performed. Therefore, the duration of immunity following natural infection is not known. In summary, the majority of vaccines are directed against the spike protein. RNA lipid nanoparticle vaccines have a high efficacy, 90 to 94%, and a good safety profile. Viral vector vaccines have demonstrated an efficacy of approximately 70%, and there is extensive experience with this vector through gene therapy. Significant toxicity is not expected. Protein-based vaccines are a proven technology and are likely to provide moderate protection. There is only a low risk that mutations in the SARS CoV-2 virus will lower vaccine efficacy. In other words, escape the vaccine. The duration of immunity remains to be determined.